And without further ado, I would like to introduce our first speaker, Dan Green from Great Wolf Therapeutics. So Dan holds a PhD in computational biology from the University of Liverpool, and has since then worked in both in academic and in industry on different small molecule drug development, both in preclinical and early clinical programs. And now he is heading the bioinformatics unit at Great Wolf Therapeutics. And I will hand it over to Dan to tell you more about this exciting story.
Yeah, thanks for the introduction, Johan, and thanks to Biognosis again for having me to speak. It feels like just yesterday I was giving the previous talk. So for those of you that were here yesterday, there's a little bit of repetition in the introduction, but they're very, very different stories. So as Johan kindly introduced, today's story is really going to be how we make use of immunopeptidomics to understand target engagement and pharmacodynamics of our ERAP1 and ERAP2 inhibitors. So really just to kick off and to start with a bit of an introduction to Grey Wolf. So who are Grey Wolf? We're a clinical stage biotech, and our approach is really to target this signal 1 component of T-cell activation. So T-cell activation requiring 3 signals. And most current approaches focus really on signal 2 and signal 3, so signal 2 being co-stimulation and signal 3 being cytokines. But our approach really is centric around antigen modulation. So we believe by changing the antigen presentation on the cell surface, you can change the appearance of that cell to the immune system. And we do this, as I said, by targeting 2 enzymes, so ERAP1 and ERAP2. So ERAP enzymes sit in the endoplasmic reticulum of the cell, and their role is in cleavage of peptide substrates prior to presentation on MHC1. So you will have proteins in the cell, they will get degraded by the proteasome, this peptide chain will then be transported into the endoplasmic reticulum. A proportion of those peptides will then be cleaved by ERAP or other aminopeptidases, and then they will be subsequently presented as most commonly 9-mers, but with some variance in length on MHC1. So ERAP 1 and 2 has both therapeutic applications in oncology. So in oncology, we can turn ERAP off to drive tumor visibility. So we drive the production of new neoantigens to the cell surface and increase the visibility of tumor cells. And similarly in autoimmunity, where autoimmunity is driven by misrecognition of self-autoantigens, we can turn ERAP off to stop those autoantigens making it to the cell surface. So we can play in 2 indication areas here, both oncology and autoimmunity. So 2 stories today. First one being around ERAP1, and then I'll follow on with the story around ERAP2, but both of them are centric on how we've used immunopeptidomics to not only drive biological understanding, but also then to derive a pharmacodynamic biomarker that we now even use in clinic. So to kick off first story, this is going to be centric around an experiment we did in HCT116 cells. So we took HCT116 cells and we treated them with increasing concentrations of one of our ERAP inhibitors. And then this is probably the only schematic there will be to any technical application of mass spec today, but just to say, we then lyse the cells, we do an IP pull down, or Biognosis do an IP pull down of pan-HLA antibody, and we're looking to observe what is quantitatively changed of these peptides at the cell surface with direct DIA. So we get a very, very nice and quantitatively complete experiment. So here in this particular experiment, we identify about almost 24,000 immunopeptides on the cell surface that we can then draw quantitative comparisons between. And you can see if you take all of these circa 24,000 peptides and ask simply what variance looks like in the experiment with a PCA plot, you can see quite nicely here where the dots are each individual sample, and the lines connect what are biological replicates within the same dose. So if you look at the top side of this graph, we've got DMSO grouped with very low concentrations of the drug or lower concentrations of the drug. And then as we increase the increase the dose of the drug, we get nice separation down PC2. And PC1 here just separates the biological replicates as we expect. So we started to ask questions about what those peptides might look like, or what the characteristics of those peptides might look like. And we started to see something very interesting. So it wasn't just that we were getting differential change at the cell surface. We were also getting particular characteristics of that differential change. So here, this heat map describes a statistical model, which is just a log-log model where you simply ask, if you take log2 abundances of your immunopeptides, and you ask, what is the linear association of the log of the dose that you've treated them with? You get about circa 2,000 significant peptides that change on the cell surface. And what is really interesting to see is if you follow my cursor here, so 0 nanomolar being our DMSO samples, those peptides that have relatively higher abundance in DMSO that then decrease with increasing concentrations of the drug, so they end up with relatively lower abundance at 10 nanomolar, are generally all those peptides that are 7 to 9 mers in length. And the opposite story applies, whereas if you track from DMSO, all of these peptides in this section here, lower abundance in DMSO samples, and then proportionally and dose-dependently increase in abundance with increasing concentrations of drug. And interestingly, all of these peptides in this block here are largely 10 to 14 mers. And this is what we now know to be the mechanism of action for ERAP1. So ERAP1 will prevent, or if you inhibit ERAP1, you inhibit the cleavage of these longer peptide substrates that are coming to the ER, and you could create a proportionally longer substrate pool, which then allows antigen turnover at the cell surface. So if you break that down, you can see this in a more granular manner on the volcano plot on your right-hand side, my left-hand side, where if you follow 8 mers to 9 mers, you've got some blue dots in there, quite a lot of blue dots. But as you increase the length, and these are all these peptides represented in this heat map, as you increase the length, you can see that we're predominantly upregulating those peptides that are longer. So really, really nice, high-dimensional data. And then often the question comes internally is how can we turn this high-dimensional data into a single score so we can do pharmacodynamics? So we started to think about how you can condense 20,000 peptides worth of information so we can generate a single value so we can start to do things like PK/PD modeling and understanding target engagement in the clinic. So here on the left-hand side, slightly different representation. So no longer this log-log model where you've got 2,000 peptides significant, but instead pairwise t-tests that are all compared back to DMSO. So you've got your effect at 16 nanomolar, 80, 400, 2 micromolar, and 10 micromolar. And hopefully what you can see here is that when we plot the length frequency of these peptides, so your y-axis here is the percentage of each of these lengths of peptides, hopefully you can see that we start to look at getting something that looks like a dose-response curve. So to make a pharmacodynamic biomarker out of this, we did some thinking about how you could potentially represent this or represent all of this information to generate something that was going to give you a single score. And it turns out what you can do is you can start to fit shapes to these. So you can start to fit shapes to these distributions, and then once you've fitted a shape, or once you've fitted the distribution to these models, you can start to ask questions of it. So you can start to say, what are the properties of each of the individual shapes that you fit? And that is what's represented on the right-hand side here. So these are the points on the right-hand side are where these data points would fall. And the lines that you see are the estimations from a model. So we're aiming really to fit this model and then start asking questions about how different that model is across each of the doses. And it turns out to work pretty nicely. So exactly the same experiment, left-hand side. We can see the increasing dose here. And now each of those individual points are a single point representation based on the length shift that we observed before. So 20,000 peptides worth of information condensed into length representation, now condensed into a single score. And what we can see is that as we increase the concentration of the dose, we can fit a nice sigmoidal curve that then allows us to calculate an EC50. Then to subsequently validate this, I mean, we've been with this quite a long time, so it's been validated quite extensively, both with genetic knockdown and everything. But another example of its validation here is a PKPD, or data from a PKPD model, where individual mice implanted with cell-derived xenografts were dosed to steady state. So once they were dosed to steady state, we then took these CDX models out of the mice and asked exactly the same question we're asking here. And what you can see is exactly as we'd expect. So you dose them to steady state, and then what you can do with this biomarker is quantify the loss or the clearance of drug over time. So for ERAP1, we think we have a biomarker that not only phenocopies genetic signatures, which is data not shown here, but can also allow us to do some PKPD work, and obviously now to allow us to understand target engagement in the clinic. So we've actually used this to, I mean, we're a good way into the clinical trial now, but we've actually used this to demonstrate our first in-human proof of mechanism for ERAP1 inhibition antigen modulation in humans. Okay, story number 2. Similar vein. So again, immunopeptidomics to drive not only biological understanding of ERAP2, but also condense it down to a pharmacodynamic biomarker. So ERAP1 and ERAP2, pretty similar, but complementary effects on the antigen repertoire. So both non-redundant enzymes, both with slightly different substrate specificities. And I'll get into this bit now more. Similar experimental design, so this was one of the early experiments we did. So this is with siRNA as opposed to a compound, but I'll show some compound data afterwards. So an siRNA titration of HS578 T cells from scramble all the way up to 10,000 picomolar. And what you can see again on the right-hand side is a really nice and clear separation between what is your scramble and then what is increasing the titration of your siRNA. So you get nice separation here across PC1. And again, we're working with about 20,000 quantitatively complete immunopeptides to make this inference. So I've put this in the same format just to show you the differences between what we find for ERAP1 and ERAP2. So again, you can compute the same log-log model, and you can get, I don't know the exact number, plus 2,000 significant peptides. I think it's close to 3,000 for this. And again, you get very, very nice clustering that illustrates the dose-dependent separation of the siRNA. You get 2 distinct blocks of both upregulated and downregulated peptides, but we get no length preference. So 2 very similar enzymes, both sit in the same subcellular location, doing different things to substrates in that ER. So no length preference whatsoever, and then again illustrated by the same thing on the right-hand side, where now if you stratify those differentially expressed peptides, you're getting no difference in the length that is between all of the upregulated and downregulated. So we started to think ERAP2 will be doing something different, but what is it doing differently to ERAP1? So we started to look further into the characteristics of these immunopeptides, and what started to become clear, and one example of here, is that if you look at the background motifs of all of your immunopeptides in this experiment, you can very clearly see what we'd expect. So you would expect there to be overrepresentation at anchor residues at P2 and the C-terminus. So this is the grooves that interact with the HLA prior to presentation. And if you look at what is upregulated, or what is increased in copy number following ERAP2 inhibition, we can start to see that there's this enrichment at P1 for these basic residues. So these lysine and arginine peptides are becoming increasingly enriched at P1. And if you look in the downregulated set, they're almost not selected for. So those that are losing copy number from the cell surface are not those with lysine and arginine, and we're driving the upregulation of basic residues at P1. So you can take this information And you can plot this quite nicely over your dose response. So again, moving away from this log-log model now where we're doing pairwise comparisons again. So you've got pairwise t-test back to your scrambled sample. And you can see that almost dose-dependently, we're increasing the presentation of peptides with lysine and arginine at the P1 position. So maybe you could start to think about already how you could use this information to look and start to model what the effect of increasing doses of ERAP2 inhibitors are. And again, if you revisit the heat map on the right-hand side and instead forget about your length annotation, but actually start to stratify by what peptides in each of these rows carry lysine and arginine at that P1 position, relative to not, you can see it is all of these that are lower in your scramble sample, but are upregulated in a dose-dependent manner. So we're pretty confident that this is a good signature of ERAP2 inhibition that is not only biologically relevant, but can also potentially help us understand pharmacodynamics. So again, concept is high-dimensional data. Internal ask is always, how can we boil it down into a single score? I jumped ahead, sorry. Just to say that this was also not only, we went off to validate this in different compounds. So these are 3 different tool compounds of ERAP2 inhibitors. So showed you the data in siRNA. Exactly the same is happening when you inhibit with the 3 tool compounds. So we're getting these that are going up are increased presentation of peptides with lysine and arginine at P1, same here and same here. And not only that, but this increased presentation of these lysine and arginine peptides, when you genetically manipulate ERAP2, you can also reverse the signature. So we knocked in ERAP2 to a mouse model, and so it's illustrated here. So you knock in ERAP2 into a mouse model, and now you're overexpressing ERAP2, and you can completely reverse the signature that we see. So now you're getting the same thing flipped around because you've got overexpression of ERAP2. So at this point, we're pretty confident about the signature. And now, this is the point I jumped ahead to before. Now we'd been asked to boil it down into a single pharmacodynamic score. So you can start to ask questions. You can start to ask what you would expect by random chance this presentation to be. So you can bootstrap various expectations from the data and ask the question, how likely should it be under random circumstances for lysine and arginine peptides to exist at P1? And then you can start to ask, how much do you deviate from that expectation? And if you do that, and what I've shown here is just the increase in abundance of particular peptides here on the left-hand side. But if you start to ask those questions on a global peptidome basis for these immunopeptides that we collect in this data set, you can again start to get single scores that are dose responsive, that allow us to fit a 4-parameter model or a sigmoid model and generate EC50 values. And with we're not quite in the clinic with ERAP2 yet, but we did go on to do some genetic validation. So we've taken biological inference that we've made from immunopeptidomics, and we start to ask the question whether or not you can inject that biological inference and whether or not it would drive any survival differences in proof of concept in bladder cancer patients. So what you're seeing here is there is a particular SNP in ERAP2 that occurs in about 25% of the population. This particular SNP introduces a premature stop codon at exon 10, so it causes nonsense-mediated decay. You don't get protein product. So we can use that SNP to understand what a genetic knockout would be in the population for ERAP2. And you can start to stratify these patients by their ability to present lysine and arginines at the P1 position. So based on each of us will have a different set of HLA types. Those HLAs will present different physiochemical properties of peptides. Some HLAs like this P1 position to be lysine and arginine more than others. And our hypothesis was that if you carry those HLAs, then you're more likely to benefit from an ERAP inhibitor. So ERAP2 inhibition will drive a greater peptidome change at the cell surface and drive recognition of those tumor cells. And that's sort of exactly what we're showing here. Right? So if you just trace these two middle lines being gray and red, these are basically saying if you don't have this optimal HLA type that we stratified on, so if you don't have these HLAs whereby you can present these lysine arginine peptides, it doesn't really matter if you carry the knockout or not. Okay? So you can be wild type for ERAP2, and you can be homozygote knockout for ERAP2, and it doesn't really impact survival. So you've got pretty close survival differences there. If instead you do have this optimal HLA type, and you have genetic knockout of ERAP2, then you have better survival. So this would be a phenocopy of what we aim to do with inhibitors in the clinic. And then the reverse is true, that if you do have this optimal HLA type and you have active ERAP2, well, we think you're getting trimming of those peptides, and therefore the survival is less. So I'm not going to read through all of this, but this is a schematic that basically illustrates the points just made. So if you're carrying this optimal HLA type, and you have an inactive ERAP2, then this is the sort of patient population that we think will benefit most from our ERAP2 inhibitors. And conversely, the same as I just said, that if you're not carrying the HLA type, it doesn't really matter whether or not you are ERAP2 wild type or ERAP2 knockout. So to wrap up and summarize, we've got two enzymes that we target. They're non-redundant, and they have complementary effects on the antigen repertoire. For ERAP1, we've used immunopeptidomics to not only understand what the biology of inhibiting these enzymes is, but also developed quantitative pharmacodynamic models where we can understand target engagement. And this target engagement is not only a binary, yes, you've engaged the target, it's completely dose-dependent, and we've been able to show dose dependency many times. And then for ERAP2, we've shown that it has no length preference, but again used immunopeptidomics to understand what its substrate specificity is likely to be, use that substrate specificity to build pharmacodynamic models where we can start to understand dose-dependent changes in the immunopeptidome, and also use that biological knowledge to start to think about how we might do patient selection when we reach the clinic. So with that, I'd just like to wrap up and say, I'm just the messenger here. Drug development is very much a team sport. And I'd like to thank all of these people in this picture. And again, Biognosis for having me to speak.